Thank you so much for joining us at Venture Cafe tonight. I'm really excited to hear from our speaker, artist Talia Greene, who I've been working with closely for the past 8 months or so during her artist residency and also preparing for her solo exhibition at the Esther Klein Gallery, which is open now until the end of March. So I'll put some links in the chat shortly where you can find more information if you want to visit the show in person. And we will be having an opening reception soon, TBD. So to give you a little bit of background, the BioArt Residency Program is a collaboration between the University City Science Center and the biotech company Integral Molecular, where artists are chosen to spend 3 months doing research alongside scientists in the lab. And then they exhibit their work at the Esther Klein Gallery, which is located at the Science Center. So we've been doing this residency program since 2017, and every artist has a unique approach and faces unique challenges. So I'm going to go ahead and pass things over to the CEO of Integral Molecular, Dr. Ben Durance. Hey, Ben.
Thanks, Angela. You can just call me Ben. It's okay. But thanks, Angela, and thanks, Talia. And happy to just say a few words at the beginning here. But we've really enjoyed having Talia join us. We've all been struggling through the pandemic, of course. But what's really unique about Talia's work is that we've used the pandemic for the silver lining, to find the silver linings and really emphasize, use it for the artwork. As a company, we study cell and molecular biology. We study a lot of viruses. We always have, from HIV to influenza, and of course now SARS. So when the pandemic hit, we had to change our artist in residence program quite a bit to be more virtual. But I think the upside of that, if there is an upside, is that we've been able to use SARS and the pandemic and vaccines as a launching point for new artist projects. The one that Talia is presenting today, the one that Laura Splan, as Angela mentioned, our previous artist in residence, also worked on. And the exciting thing for us as scientists in the company is that the art is the science. And as Talia will present, it's not even art that's inspired by science. It is science. The vaccine, the viruses that have been created, or vaccines in this case, mRNA vaccine, that have been created, is the artwork, is the piece. So we're really excited about that, and many of our scientists have gotten very involved in working with Talia, pointing her in the right direction, providing resources, providing information, and being directly involved as artists, as scientists, to help Talia fulfill the vision of what she's brought. So we're really excited to see the exhibition. I would definitely encourage everyone to stop by in person. I'm sorry we can't all be there in person today, but we will have a reception at some point and get to meet everyone and see all the artwork in person together. But thank you, Talia.
Yeah, thank you so much. I think I'm just gonna get started. So, well, those of you who were here last time when I gave the talk at Venture Cafe know that I'm hoping very much for no Zoom screen sharing snafus, and I think we're good to go, right? Everyone, awesome. Okay, so let me just get my screen organized here a little bit so I can see everything. Okay. So I just wanted to start out by showing you some installation shots of the show to give you an overview of the 3 main projects that I developed during my residency at Integral. So on the left, I know my cursor is pretty small, I'm going to be pointing, hopefully you guys can see, but on the left is a piece called Antisense Therapy, and it's a sequential piece that shows the development of the code for an mRNA misinformation vaccine. And then here you can see the vaccines displayed on a different wall. And then in the back is a piece called #Delta!! That is a series of prints and also a single channel video. For those of you who are going to visit the show, it's in this room here. And then there's a piece called Internet Hazard, and that is a sound and print installation. So since I gave a talk about some of my other projects last year at Venture Cafe, I thought tonight we would just take a deep dive into the process and collaboration that led to these specific works. So for those of you who are not familiar with my work already, many of my projects involve a lot of research, and my interest in science has been more related to the intersection of social and environmental histories. So this bioart residency definitely opened up a very different line of study for me. I knew that in order to really engage with the scientists at Integral, I needed to have a basic understanding of what they do. In other words, I had to try to speak their language. So one of my first steps was to buy Molecular and Cell Biology for Dummies. While not knowing much about science could have felt like a liability, I decided to capitalize on my ignorance and make it a main theme of the residency. Because after all, as you all know, one of the main issues during the COVID pandemic has been the myriad ways that science has been misunderstood, but also not clearly communicated. So this is the first page of my notebook where I'm starting to think about some of the questions which would become themes for the project. So some of the themes about translation, how is science communicated to the public? How is it interpreted or misinterpreted? And then also some questions about the role of artist and interpreter and what happens when artists and designers communicate science. So while I was trying to learn some of the basic science on my own, at Integral, I began having conversations with the staff, really just to see what they had to say about these general questions. And to be honest, I could tell that some of them were confused about what I was doing, and they were having trouble seeing how our conversations would lead to art. But at that point, I was not actually concerned about the art. I was just interested in hearing what they had to say. And one of the themes that kept emerging was about the internet and disinformation. So that became my second avenue of research. And for me, it was almost as alien as the science. As you all know, the internet is full of conspiracy theories and misinformation related to COVID. We're all too aware of that by now. But it was very easy to find a full range of misinformation, from the completely ridiculous, like this one, to distortions of science and everything in between. So one of the conspiracy theories that seemed to be of particular interest to the scientists was related to questions about the origins of COVID, and in particular, the lab leak theory. So we now know that there's evidence that the virus could have escaped from a coronavirus lab in Wuhan, but at the time, this seemed to be just another conspiracy theory. But as I was doing some of this research, I organized a group of Integral employees who are from China and also Americans of Chinese descent to talk about the diplomatic rifts that have been escalating and also the backlash that Asians have been experiencing in this country during the pandemic. So as I was following all of these different avenues, I started asking myself questions like, how do we pick and choose what we want to understand or want to hear or believe? And that led me to make these little poems like this one that you see here. That was really just me rearranging the words to the terms that I was learning. And these little word sketches led to questions like, how does meaning change if small things are rearranged? How does something that makes sense to one group and make no sense to another group all of a sudden become meaningless for everyone? So I also started looking at and rearranging some of the letters used by microbiologists, such as A, C, T, and G, which refer to the nucleotides that comprise our DNA. And as Ben mentioned, this residency happened during the pandemic. Of course, we're still in the pandemic, and so most of the conversations happened over Zoom. And so this is a screenshot of me explaining some of my ideas to Brianna Tyrell. And you can see a little picture of Brianna in the corner. So after I explained some of the things I was doing, she explained the way that she uses the DNA sequences in her research. So I wanted to invite Brianna now to tell you a little bit about what she does and what you're seeing here in this image.
Hi, so my name is Mrna. I'm the molecular biology project specialist at Integral. In this image here, I'm actually showing Talia a way that we can visualize DNA sequences to look at the purity and also confirm the sequence of the DNA. So what you're looking at here is those A's, C's, T's, and G's. You can see each of them have their own color in these peaks, and we're aligning them to a known sequence to check that it is what we think it is. This is important here because you can see these A's, C's, T's, and G's can be rearranged, like Talia said, to kind of make different things. And on the next slide, specifically, A's, C's, T's, and G's are these nucleotides for DNA sequences encode amino acid sequences, which are represented as these different letters here. So this chart here is a way that we can visualize and break down kind of those 3-digit codes and how they can come together to create something else, the amino acid sequences. And we see how this can kind of grow even further to create proteins, but also on the right here, we have specifically the spike protein for the coronavirus. So something that we kind of talked about is how we can kind of take this pseudo alphabet, which you string together to make things such as proteins, and turn it into something else. So Talia was able to take these amino acid codes here, use it as her pseudo alphabet, and then kind of write her misinformation spike protein that I was then able to translate back to those A's, C's, T's, and G's so that we can actually synthesize it in the lab.
Yeah, and so I will show you the product of that now. So basically, at this point in the residency, I was bringing together all of these different ideas that had been gestating, my conversations with the scientists, my research about misinformation. And so at that point, really after my conversation with Mrna, the one that you saw, I started embedding the misinformation that I was finding on the internet within the spike protein amino acid sequence. So I used excerpted phrases like this one that you see here, I have an immune system, no vaccine needed, and spelled them out using only the letters that are found in the spike protein code. So in this phrase, the O is missing from the no vaccine needed because there's no amino acid represented by the letter O. And when I took a letter from the spike protein and used it for the misinformation phrase, I also added an underscore to the original sequence to show that then that was missing. So that's why at the end, some of the sentences are harder to read because I was basically running out of letters to use. So the idea was that over time, the original sequence of the spike protein would become mutated and distorted by the misinformation, becoming something entirely different from its original form. So at this point in the residency, I was starting to find some visual answers to some of my questions about language and communication. Questions like, how does science get distorted? And what is the role of the internet and social media in these distortions? But I was also talking to other scientists from the lab about what they were working on. And so another scientist who was very helpful as well throughout this process was Hayley Crawford. So I'm going to introduce you now to Hayley Crawford, and she's going to tell us a little bit about what you're seeing in this slide here. So Hayley, welcome.
Thanks, Talia. Hi, I'm Hayley. I'm a virologist at Integral Molecular. What you can see here is a microscope image of hamster kidney cells, magnified 10 times. We use these cells in the lab as factories to produce viral particles that we call RVPs. And these viral particles can be used safely by scientists studying viruses or developing vaccines against viruses because the particles aren't capable of more than one infection event and they can't infect people working with them. So they are infinitely more safe than working with live viruses. And over the past 2 years, we've used these cells to make almost every SARS-CoV-2 variant into RVPs, and vaccine companies use them to see how effective their vaccines are against all of the coronavirus variants. Oh, and this photo shows the same cells, but looking through a filter. There's Talia sitting at the microscope, you can see that these cells are glowing with green fluorescent protein, which is a protein from jellyfish that we use. And this indicates to us which cells the virus entered. So the fluorescence is a great tool for us to be able to visualize an otherwise invisible process. It also allows scientists to figure out which antibodies can neutralize SARS-2, neutralize as in stop infection, because a great antibody would mean that you would see fewer green cells, which means fewer infected cells, indicating an effective antibody.
Yeah, so thank you, Hayley. Yeah, I mean, as you guys are seeing, I learned a lot of science during this project. So yeah, Hayley initially just showed me some images of the cells, and we talked about how they're used, and then she invited me in to come and photograph some of the cells under the microscope. So that's what you're seeing here. And then at the same time, I was talking to Hayley, I was talking to Mrna, I was still doing the research, and I came across this image, which is a grid of DNA nanostructures that can be programmed to form specific shapes. So, and you can see some of them are pretty silly, quite whimsical. You can see smiley faces, you can see letters, numbers, and then up here, I don't know if you can see this, this is a heart. And the heart reminded me of social media icons that help spread misinformation. So I began to create these sequences from Hayley's cells, which would gradually come together to form the 4 Twitter icons. So I used the sequences then to create a wallpaper pattern in which the cells are mutating and forming different shapes, and they're glowing greener as they form the icons. And that green, of course, references the GFP, the green fluorescent protein that Hayley was just mentioning. So while the icons in this piece represent the role of social media in spreading misinformation, icons and symbols in general are also a great example of a place where design and communication meet. They're also an example of a visual and universal language. So on the right is a photo from the lab, the Integral lab, that has one of the many biohazard signs. And actually, I took some of the biohazard photographs when I first went there, but then I accidentally deleted them. So I asked Hayley to take one, and she took a bunch. So I could have shown you many, but there's this one. So that's the lab. And then on the left are some of the icons and signs that I took inspiration from to create this internet hazard sign. It says, "Step here for your safety." So this is the installation view where you can see where I placed 2 of the 4 internet hazard floor signs. So there's one on each side of a cube. Those of you who have been to Esther Klein Gallery, you know it sits in the center of the gallery, so you can walk around the cube. And then the floor signs invite viewers to step here, but it's ambiguous. It suggests it's for the viewer's safety, but it's not really clear what they're trying to avoid. So standing on the spot activates a sound piece, which combines audio collected from the internet relating to COVID-19 misinformation, but also the polarized arguments about masking and vaccinations that we're all familiar with now. So on the back wall of the cube is a shelf containing vials of the mRNA vaccine that Ben mentioned when he introduced the project. So this was developed at Integral as part of the residency. And using the final mutated sequence that contains all of the misinformation phrases, we basically created this vaccine, which theoretically would work just like the COVID mRNA vaccines. So I invited Joe Stafford now because he was the person who actually made the mRNA vaccine. So you can see his hands here. And I asked him to talk a little bit about how mRNA vaccines work and what his process was. So Joe.
Thank you, Talia. So I'm Joe Stafford. I'm a research scientist at Integral Molecular, and I made the misinformation mRNA vaccine for Talia. So the figure on the left-hand side of the screen is just a basic description of what the mRNA vaccine. It shows a spike protein as the orange thing under the microscope at the top is the surface, is what's on the surface of the coronavirus, which is ideal for making the vaccine. The spike protein on the virus is what binds to the cell surface that the virus is trying to infect. So that's what's going to bind to the cell. Then the vaccine delivers the mRNA, or the genetic code of the spike protein, to the vaccinated individual's cells, teaching the host cell how to create their own spike protein. And then this process triggers an immune response in which your body then creates antibodies to fight off the COVID-19 infection. So on the right-hand side of this, the right-hand side of the figure, this is how I actually made the mRNA vaccine. So once I got the misinformation vaccine from Mrna, I basically took the DNA and performed a process called in vitro transcription to make the mRNA. The top photo shows me making the mRNA, and the final mRNA can be seen in the top right photo. In the bottom photos, you see me preparing reagents to visualize the mRNA. So once the mRNA was visualized and validated, we encapsulated the mRNA into a lipid mixture that could be used for immunizations. And that is what you can see on the display in the gallery.
Awesome, thank you, Joe. So this is the installation view of the final vaccine that lights up and emits this sound when a viewer stands on the yellow floor sign. And I'm just going to play a short excerpt because it's a minute or so, and I want to encourage you to all come to the space and listen to all of them in person, but this just gives you a sense.
The COVID-19 vaccine is not a live virus vaccine. So it is not possible to shape the virus because of the vaccine.
It's actually contrary to all the rules.
Talking about an unproven, untested vaccine. It doesn't even seem to really protect people.
It's actually
because
contrary to
people
all
who
the
got the
rules.
vaccine are getting sick. The best things we have is our own God-given natural antibodies.
It causes the immune system to actually fight the virus strong.
That was just a little excerpt. So the sound pieces start to address the question at the bottom of this page, also from my notebook, you're getting used to seeing that, which is what are the consequences of misinformation and not understanding science? And the sound is a cacophonous, repetitive din of information, some true, some false, some misleading, which becomes all jumbled together, and it's almost impossible for really any of us to unravel. But as I mentioned earlier in the talk, the COVID-19 pandemic has not just highlighted the role of language barriers between science and the public, but it's also heightened barriers to understanding between countries, and in particular between China and America. At the same time, we're all being forced to learn a little bit of Greek with every new variant that comes out. And on the right, you can see a sign in the lab displaying the Greek alphabet, which they use to label flasks. So my last piece began with research on the letters and symbols in a range of alphabets, including Greek, Chinese, and English. So on the left are some of my many notes that were exploring both the form and meaning of the various characters. And I mentioned earlier that I met several times with a group of Chinese and Chinese American scientists for the project. And one of those scientists was probably the person who I spoke to and met with the most throughout this project. And these are some of the text exchanges towards the end of the project, but we also chalked up many, many hours with him just basically teaching me Chinese and helping me to choose which characters for you to use for the next piece that I'm going to share for you. So you can see, again, this is toward the end and we got really specific. We were thinking about the individual meaning of each character and also how the meaning changed when adjacent to other characters. So each character or symbol is composed for this piece of a different layer of the same cell. And I made the Greek letters out of one layer, the English symbols out of another, and then the Chinese characters out of a third. So now I'm going to turn you over to the last scientist, we're almost done here, Riley Payne, who photographed the original cells for me. Riley, welcome, thank you.
Yeah, thanks, Talia. So this technique is really similar to what Hayley already described. But in this case, the glowing comes not from any activity of a reporter virus or infection or anything like that, but from specific stains that we can use to label the different parts of these cells. So the cells on the left are really, that's kind of the cytoplasm, the guts of the cell, what's inside. The center is the nuclei of the cell that contain all of the DNA. So that's really DNA that you're seeing inside cells. And on the far right there, that's when we label just the outer surface of the cell. So you can really see how there's kind of, highlights near the out. So, we're able to do this, and we use this technique really mostly for this exact application, is to identify these subcellular structures and know what we're looking at when we use microscopy.
Right. And I would just add that when they break down the cells, each layer is a different color, right? One is green, one is red, and one is blue. Is that right?
It is, and it's all based on kind of different wavelengths of light that the different stains emit. But in many ways, in the microscope, we label it different colors. They're different wavelengths, but they all show up as white in our images. So they're often pseudo-colored by scientists, but in essence, any coloration here is equally accurate for all of these different things. So that's cool.
Thanks, Riley. So this is the installation view of the final configuration. And as I mentioned before, I was attracted to the formal similarities between the different characters and how making a slight variation to the shape would completely change what it was. But I was also interested in the overlapping meanings of the characters in multiple languages. For example, the equals is also the number 2 in Chinese, and the chi, which is the 14th letter of the Greek alphabet, is 3 in Chinese. And then other examples that are more familiar to you, like the X, which is also used as multiplication in math. So I was just really interested in just discovering all of the sort of layers of meanings behind these symbols, but also what happens when you sort of put them together in different configurations. So this is just sort of a map that shows you the meaning of the different characters, and some of the orders of the characters make no sense, but then some combinations do create actually a legible phrase. So like with this one here, that says country is greater than or equal to dust. And some combinations require more of a sort of creative interpretation or context, like delta is greater than or equal to hashtag. And then other phrases require even deeper cultural knowledge. So the combination of the characters Chen and Tu can mean life is like dust. Those are some of the many things that I learned from the Chinese scientists at Integral, who I mentioned earlier. So in order to emphasize this theme of subjective meanings, I created this video for the show. And so this is the last piece I'm going to show you. I'm not sure if I'm going to play the whole thing, but I'll probably just start with a short piece and a bit, and then I can answer any questions you have.
In Chinese, the number 3. The New Testament has God declaring himself to be the Alpha and Omega, the first letter in the English alphabet. In Chinese, the number 2 is a variant of SARS-CoV-2, the virus that causes COVID-19. Beta, the second letter of the Greek alphabet. In Chinese, the number 3, the 23rd letter of the Greek alphabet. Gamma, the third letter of the Greek alphabet, the third in a series of items, categories, etc. Originally derived from estimates occupying the middle delta, the fourth letter of the Greek alphabet, a triangular tract of sediment deposited at the mouth of a river, typically where it diverges into several outlets. Began to spread quickly by mid-December, around the same time as a code word representing the letter D. This increase is thought to be at least partly because of one or more mutations in the virus. A difference between two things or values. The variant is also notable for having more mutations than normally seen. The discriminant of a quadratic equation. The Delta variant is highly contagious, more than 2x as contagious as previous variants. Some data suggest the Delta variant might cause more severe illness than previous variants in unvaccinated people. Lambda is the 11th letter in the Greek alphabet. In astrophysics, lambda represents the likelihood that a small body will encounter. Alpha refers to a dominant person or their behavior, especially with respect to socially aggressive, hypermasculine men. Critical caring to die that are people infected. The Chinese character for person. The beta variant, also known as BE1. The Chinese characters for the people. Viruses like SARS-CoV-2 continuously evolve as changes in the genetic code. The Chinese characters for people of a certain nationality. Deltas are important in human civilization, as they are major agricultural production centers and population centers. Earth country or sovereign land. Dust. 10. One speck of dust from an entire era may not seem like much, but when it falls on your head, it's like a mountain crashing on you for life. Consists of 2 protons and 2 neutrons bound together into a particle identical. The 15th letter of the Greek alphabet. In ecology, site survival probability, or the probability that a species will continue to occupy a site if it was there the previous year. The term beta refers to advice on how to successfully complete a particular climbing route, boulder problem, or crux sequence. The Roman numeral 2. And around 50% more likely to require. In written mathematical logic, the tilde represents negation. 生存还是毁灭，就是那个问题。That is the question. The final letter in the Greek alphabet, omega is often used to denote the last, the end, or the ultimate limit. In actuarial sciences, used to represent the maximum lifespan that characterizes a mortality table. In contrast to alpha, the first letter of the Greek alphabet.
I decided to show you the whole thing because we have so much time left. So just to wrap it up, I just want to thank all the integral staff and especially the collaborators so much. It was just an incredible experience. I learned so much and had so much fun. And thank you also so much to Angela, who just put up with so many ups and downs throughout the installation process. And then I also just want to acknowledge 2 members of my family, Amit Greene and Mark Greene, who helped me with the software development, the hardware engineering, the 3D fabrication of the sound installation. And so it is an interactive sound installation, and none of that could have been done without them. So thanks to my family as well. And thank you all for coming. And I guess I'll stop my screen sharing now and open it up in case anyone has any questions for me or any of the scientists, really.
I just wanted to make a comment real quick. It's been such a pleasure working with you, and I'm so impressed by how many layers this project has. You really took like this deep dive into the material, and your exhibition is so well thought out and very creative. And very relevant and timely while also having a great sense of humor. So I just wanted to congratulate you on the culmination of this research that you've done. You did an amazing job. So
thank you, Angela. Couldn't have made it through without you. And a lot of other people as well. Definitely got a lot of support through this. Anyone have any questions or?
I had a question.
Laura? Oh, I wasn't sure who said. Yeah, thanks.
I was wondering if you could talk a little bit about the creative approaches you took in these pieces, and especially the procedural art and the use of the kind of embrace of chance operations and modularity. And I'm just wondering how much of that was inspired by the science you were looking at and the laboratory techniques you were learning about, and how much of it felt like a seamless continuation of the way that you've approached other projects like the redlining project, and or if you kind of adopted, if some of that was sort of a pragmatic response to.
Yeah, that's an interesting
residency.
It definitely, the archives project was definitely its own beast. But I guess now that I think about it, I think so Laura, I just want to introduce Laura because she did this residency, but we also went to graduate school together. So she's known my work for a long time. So she's probably noticing some things that maybe some of you are noticing, which is a lot of, I guess, similarities. Even though the projects are really different, I think that there are sort of approaches that I tend to default to, whether I'm aware of it or not. And a lot of it, the sequential thing, I tend to do a lot. And I know there are also some people who like Mark Campbell, I saw you there, hello. He was the chair of the Department of UArts when I first started teaching at UArts and taught a class called Time and Motion. And I think that was one of the first times that I started to think about the power of the sequence as a way to express time passing. And I've taught it a lot and incorporated it into my work a lot. So I think that idea of the sequence is sort of a modality that you really see in a lot of my work, and it is in the archives project and earlier projects as well. And I think also typologies is another, I guess, modality that is something that has come up a lot. And I think I mentioned at the beginning that science hasn't been really hard science. It hasn't been a huge focus, but I definitely have been interested in sort of the periphery of science. And I think the idea of sort of typologies is probably taken from that. So I think that's probably another sort of thread that runs through my work. I don't know if that answers your question, Laura, but yes. Thanks for asking a question. Yeah.
Any plans to move it to other venues?
No, I think it's pretty site specific. I think it just has to stay here and then, or at the Science Center, and then be taken down.
I didn't realize the significance of was it Omega that you highlighted. I don't know where that is in the sequence of Greek alphabet, but it makes me a little bit nervous when we get.
I know. And my husband, Mark, kept saying, you have to add Omicron, you have to add Omicron. And I knew he was right, but somehow I just couldn't. And I think it's probably for that same reason of just but then it would just be endless, it would just be one Greek letter after another. And we just stop it here, or just have it be this moment in time.
Also, Ben, I think Omega, as Talia showed in that video, is like the final one. It's the last one. And that's why I think no variant has been called Omega yet, because it's not the last one. And also, funnily enough, there's a book by Michael Crichton called The Omega Strain, which is about a virus that wiped out all of humanity.
Yeah. Let's not get to Omega then, right? I guess if no one has any questions, thanks all so much for coming and listening. And I hope please come see the show.
Thank you, Talia. This was a great talk. And I just want to echo Bob's sentiment. What did he say up here? This talk is great. Learning about the art and the science at the same time is brilliant. I love the way that you structured this talk, and I loved how all the scientists participated. So thank you so much, everyone from Integral, for coming tonight.
And thanks again for all of you for hosting and helping bring this project to fruition.
Of course.
Yeah, thanks, Talia. This is wonderful. And everyone, yeah, definitely come by and see the exhibit in person. I think it's up for the next 2-3 months. Is that
Till the
right?
end of March. Yeah.
March. And hopefully there'll be a reception where everyone can get together in person.
Yeah.
Definitely.